Hello and welcome to the second episode of this yet-to-be-titled series. Today the study we'll be covering is about protein synthesis in relation to neuronal activity. So, first, some context. The way that neurons communicate, if you remember from the last episode, is in structures called dendrites that are located at the head of the cell. And protein synthesis refers to the act of synthesizing, that is to say, making a protein. Let's start this then. Activity-dependent protein synthesis. When a neuron activates, it starts rapidly producing proteins in its dendrites. This is regulated by a gene known as eIF4G2. The reason that it's named this is because eIF4G2 stands for Eukaryotic Translation Initiation Factor 4-gamma-2. The full details of this name aren't important, but what does matter is that this gene binds to RNA in order to control the process of translation. The process of translation is essentially when RNA, which is one form of genetic material that can be found inside of a cell, goes through a structure within the cell called a ribosome. That ribosome reads the RNA and makes a protein, slowly assembling it from instructions through the RNA. So what IEF4G2 does is binds to the RNA and controls what parts of that RNA are being translated, which therefore controls which proteins are being made. So, the actual experiment that they did is that scientists used certain chemicals to make the neuron imitate firing, having electricity run through it. They followed this up by using two methods to tag certain proteins, as well as identify which genes were being translated. These two methods were called TurboID, which is the one that tagged proteins near particular parts of the neuron, and CLIP, which were the ones that identified the RNA being translated. What they found was that different proteins were present and different genes were expressed, depending on whether the cell was at rest, not having a signal fired through it, or active, having a signal fired through it. This study was important for a couple of reasons. For one, since memories are currently thought to be encoded in the connections between different neurons, this could shed some insight into how memories are formed. Because the fact that there were different proteins and different genes being expressed whether the neuron was on or off, essentially, means that there is something that happens within a neuron, depending on whether or not it's communicating with other neurons. This could also, for similar reasons, relate to the process of plasticity within brains. Neuroplasticity is essentially the process by which the brain changes itself, forming and getting rid of connections, creating new neurons to replace old dead ones, and etc. Since this effect is connected to, again, neurons communicating with each other, there could be an element here that's related to plasticity and how the brain learns things and manages to accumulate that learned information into neurons themselves. The sources are in the description below.